Hello and welcome to this H3 Africa session, which is report back from the working groups. We've got 8 working groups reporting back in this session. They'll have 8 minutes each, and some of them are pre-recorded and some of them are live. So welcome to all the attendees. If you have questions, please post them in the Q&A box, and we'll have a Q&A session at the end of this session. Any questions that aren't answered will still be answered after the session at an appropriate time, so you won't miss out. Anyway, so we'll start off with the first report back, which is the Ethics and Community Engagement Working Group, and the report is by Arisa Sabakaki. Thank you.
Good afternoon, ladies and gentlemen. This is a summary of the Ethics and Community Engagement Working Group session that was held a week ago, and this is a summary of what transpired in that meeting. And on the agenda, we had 3 items: benefit sharing in H3 Africa projects, community engagement during COVID-19 pandemic, lessons learned, and any other business. The Ethics and Community Engagement Working Group is preparing a manuscript on benefit sharing in how benefit sharing is implemented in the H3 Africa project. Therefore, we gave 2 questions to members of the working group and principal investigators to give us information that would inform the manuscript we are writing. And the 2 questions were, question 1 was, which derivative of your research do you share or are you planning to share with research participants and with or their families? The second question was, are research participants compensated in your project? If yes, what do you compensate research for and how is the compensation commensurate? We gave the PIs a definition of benefit sharing based on Schrader et al. 2007. Benefit sharing is defined as the action of giving a portion of advantages or profits derived from the use of human genetic resources to the resource provider to achieve justice in exchange. Benefit sharing may also be in the form of compensation. So for question 1, only 2 projects were returning relevant primary genetic research upon request by parents to help improve adjust care. And no project was returning incidental findings. Projects were not so sure how to go about the return of these results. However, members indicated that the return of individual genetic results should be directed by the H3 Africa feedback of findings policy that was developed by the Ethics and Community Engagement Working Group. It's available on the H3 Africa website. Only 1 project is preparing for community feedback to provide participants and communities with aggregate data and discuss the disease mitigation measures with stakeholders. It's very important that if results are returned and are told there are problems, mitigation measures have to be in place. Otherwise, you don't return results and leave participants to fend for themselves. Community engagement was seen as key in the return of these results and feedback to individuals and communities. And some projects have ongoing educational outreach programs. For example, there is a project that has an ongoing education outreach program to inform community about visual loss. Concerning question 2 about compensation, all projects are compensating participants for time inconveniences and effort, and they are reimbursing participants for their study, for the study-related costs incurred. About community engagement during COVID pandemic, lessons learned, most projects have migrated towards virtual platforms. Some of them had suspended community engagement activities because of the total lockdowns in several countries. For example, in Eswatini, the CAFGEN project has completed a nationwide genomic public engagement and have received feedback on people's future learning preferences, and this has been mostly done using online platforms. Some projects, basically, like I've already said, had to pause community engagement activities, but now in most countries, these lockdown restrictions have been either removed or have eased, and now projects, community engagement activities are picking up, and projects have had to reduce on the number of participants during community engagement, especially if the activities are indoors. And this is done so that it's in line with the guidance from the government from the different countries. I would like to acknowledge the H3 Africa Consortium, the Ethics and Community Engagement Working Group, and the funders of H3 Africa, the Wellcome Trust, National Institutes of Health, and African Academy of Health Sciences, Thanks very much for listening to me.
Thank you very much for that first presentation. I should have said at the beginning that when attendees post questions in the QA session box, please could you indicate who the question is for, because we have a lot of different topics being covered in the session and multiple presenters. Anyway, so we'll now go on to the next one, and we're going to have a slight change of schedule and move on to the cardiovascular disease working group. And the leaders of this for the presentation are Michelle Ramsey and Bamidele Tayo. Thank you very much.
Hello, this is the report back for the CVD working group. My name is Michelle Ramsey, and I'm the chair of the working group, and Bamidele Tayo is the co-chair. So first of all, who are the members of the CVD working group? We now have 8 different PI research groups who are part of this working group, and you can see them here: kidney disease, cardiometabolic disease, cancer, stroke, diabetes, heart disease, glaucoma, and sickle cell disease. So first of all, the publications that have emanated from this working group. So our data resource profile called CHAIR, which was published in 2019, and as you can see here, has already had 12 citations. And then our proof of concept that we can work together, which was a paper on regional patterns of variation in terms of the association between obesity and hypertension in different African contexts. That was published in Hypertension in 2020, and that has already received 10 citations. Then in terms of sustainability, we have been looking at grant applications, and this particular grant application was done together with BioNet and with the Genome Analysis Working Group. And this grant has now been awarded and has resulted in the PRIMED Consortium, which is polygenic risk score methods and analysis for populations of diverse ancestry. And what we are adding in terms of the cardinal study site is data from Africa. So you can see that this project started in 2021, and it's a 5-year project going up to March 2026. So the principal investigator and reporting investigator is Sally Adebamowo, and Bamidele and I are multi-PIs for this grant, but of course it will involve the many people who contributed towards writing this grant. So from the H3 Africa, the involvement is for these groups shown here, but also includes, but the application also includes other people with cohorts from Africa, and then also replication cohorts. So we've also been involved in applications to the HHERE, which is Human Health Exposure and Analysis Resource. And that is a consortium that will do typing on existing samples provided that they are related to environmental health and have some scientific question in mind. So Abidjan made application, which wasn't successful in the first round, but we're busy revisiting that application, having now had good conversations with them. And the kidney and acne groups have also indicated that they're interested in applying to HCare for genotyping of some of their samples. So some of the important things we learned through our interaction was that they don't have the capacity to do many thousands of samples, and would put the limit at about 1,000 participants. They also need a specific question related to a health outcome. And what is very important is that when engaging with them, one would need to agree to sharing individual level data with the consortium, and they will make it available to others who have a bona fide academic affiliation. Then our objectives for the next 10 months or a year are that we want to add more information and data to the Chair REDCap resource, and we want to focus specifically on publications and knowledge generation. So there's sort of 6 things that are important to us. So the first is that we understand that every project has their primary objectives, and that whatever we do together should not jeopardize those. Then the projects that we propose as the CVD working group would be projects that can't be done by any one of the 7 or 8 groups alone, but only in terms of collaboration between 2 or more of the groups. Then we understand that we have a limited window of opportunity to work together, and that all participation is voluntary, and that we encourage young and established members of our groups to be part of the planning, analysis, and writing of the papers. And then also that this is an open forum where anybody can suggest analysis using the Chair resource and the genetic data, and could indicate where they want to lead projects. So these are some of the projects in progress. So the one is led by Timothy from the Kidney Group, and that is looking at association of albuminuria and decreased GFR with cardiovascular disease in Africans. And here the potential groups that can participate are kidney, siren, diabetes, and Abidjan, because they already have data on the biomarkers for albuminuria and GFR. The next project is looking at the association of ApoL1 risk variants with blood pressure and albuminuria in adults without chronic kidney disease. And this will be led by Jimoa Adu and his group. And here we know that the kidney, Abidjan, and siren groups can participate, and hopefully the Eyes of Africa will be able to participate if they generate their genetic data this year. So you can see what will be needed there, and very limited genetic information for the ApoL1 risk variants. Project 3 is about GWAS for anthropometric traits in Africans. And this is being led by the Abidjan group and could potentially involve kidney, Abidjan, siren, and acne, and Eyes of Africa if the timing were right. And this would require either genome-wide genotype data or summary statistics done individually for the traits shown. Project 4 is GWAS for blood pressure traits in Africans, and basically is the same as the previous one, except for the phenotype outcome. Potential project 5 could be led by the siren group, which is really looking at the prevalence and indicators for stroke in sub-Saharan African countries. And at this stage, only siren and Abidjan have the full sort of stroke-free questionnaire, but there is limited data from kidney, diabetes, and acne that could potentially contribute to this project. So there are a number of challenges for this working group. So first of all, the Chair resource in terms of the REDCap tool only has 2 data sets at present. So we need to work with each of the other groups to have more data uploaded into the REDCap. Then data analysis challenges include that the fact that each of the studies was actually designed for a different purpose, and therefore putting them all together in analyses is complicated and needs to be explained very carefully, because when we look at the cases, they are different phenotypes depending on the study. And also when we define controls, that is complicated because in the disease studies, the controls are often disease-free for that specific disease, but we also have Awigen, which is a population cross-sectional control. Then there are several time challenges. One, that we know that H3A is closing out, but also the genotyping that hasn't yet been done for the diabetes group, the Eyes of Africa or Sickle Gen Africa. And then, of course, the fact that it takes a while to do the analyses and interpret the data and develop the publications. And then finally, also our challenges in understanding how best to share genomic data and or summary statistics and how we can work with each of the groups in terms of what they would be more comfortable with. So in terms of the next steps, it is about uploading more data into the REDCap for chair and working towards our publications. And Bamidele will be happy to answer any questions that you may have. Thank you very much for your time.
Thank you very much for that presentation. That was very interesting about the potential projects and challenges. We'll now move without delay to the next presentation. We're going to have a phenotype harmonization working group report back next from Catherine Johnston. Thank you.
Greetings to the H-Africa Consortium members. I will be giving feedback on the Phenotype Harmonization Working Group activities and our most recent session. My name is Catherine Johnston, and I'm the chair of the working group alongside Lyndon Zass, our co-chair. These are the activities that our working group has been busy with and some of the interactions that we're having. And in our most recent session, we also discussed sustainability and plans for the final year. I'll start off with the Phenotype Standards Project. This is headed up by Lyndon and has involved a number of HRA Bionet members and HRA Africa members contributing to developing standardized data collection modules in various areas of research, all within the umbrella of genomic research, and providing projects with a way to harmonize their data prospectively. But they can also be used for retrospective data harmonization to find common data models to harmonize to. So these modules have been developed based on kind of minimum recommended variables to be collected in a standardized format. They map to ontology terms, which are internationally recognized standardized terms, so that they make it much more interoperable and they make sharing and combining data much easier. The plan is to publish and share these back to Phoenix as well, alongside with publishing them on various platforms. At the moment, the COVID-19 module is available on the HRA Bionet GitHub repository, but the other modules will be soon to follow suit, I think before the end of this year. Along with these phenotype standards, standardized modules, they have done a lot of work on developing comprehensive guidelines for the data that is collected and recommended for collection. It provides data dictionaries, a REDCap implementable project file so that you can set up a REDCap clinical database very quickly using any of the modules. And there's also work underway for developing paper-based forms for data collection. The ethno-linguistic ontology was developed to address difficulties defining population and social groups when publishing population genetic data. This is a really essential resource for standardizing the terminology used, and it's needed quite extensively and internationally to define African populations. A lot of work has gone into this ontology, and at the moment they're just dealing with some technical difficulties getting it into an online web interface. Once that is resolved, the hope is that it will be made available for further review, and then that studies will be allowed the option to include the ELO terms into their phenotype data sets. There's plans to develop a REDCap example database using the phenotype standards and core phenotypes because we recognize that they could be a bit intimidating for use. And so the intention is to use the rare diseases module as a use case alongside those core phenotypes, and also release this with a set of how-to videos for data managers to step through building a longitudinal clinical database. The HRAF Bionet Phenotype Harmonization Project has pulled together a number of members who are involved in harmonization efforts in Africa and across international organizations. So some of the key efforts have been work in the chair resource, mental health task force, Seneca, and sickle cell disease. And the main output of this project is to work on a manuscript paper providing phenotype harmonization key considerations for genomic research. And this stems from many commonalities, experiences, and challenges that a number of members have learned from in these various harmonization activities. So the project members have also built SOPs and guidelines along the way for harmonization steps that are needed to be taken, and we recognize the importance of imparting these to the Africa Consortium as soon as possible. So when it comes to other interactions for our working group, these have been with the mental health task force. Lyndon has worked extensively with 3 core projects within Africa that are collecting mental health data, and they've developed a common data standard for projects. They've also identified 8 other projects within Africa who are collecting data that's pertinent to mental health. This is by emailing PIs and also through attendance to meetings and discussions. And at the moment, 2 of the projects have mapped their data to the common data template developed. And a 3rd project is still collecting data, but will also be mapping their data to this model. And the plan is that they will then be able to prospectively prepare themselves for data harmonization. The microbiome working task force has also been developing a common data standard with Lyndon's assistance for harmonization. And so their plan is to conduct a final review before they deploy it for testing. And this common data standard could be used by future studies for upcoming grants. The cardiovascular disease working group is probably the most advanced in terms of its harmonization activities. And I've been working with that working group to develop the chair resource after they established a set of common phenotypes. This was done prior to the standard series release. And since then, we've developed a data transformation script and a mapping process for the working group studies involved. And 2 of the projects have their data imported into the chair resource already. There have been some challenges, and that's namely being slowness in getting clean data sets available for transformation, some internal harmonization that was required in projects first, and then establishing a clear process for validating that transformed data. Most recently, the Genome Analysis Working Group has put out a call for a number of phenotypes that it would like to harmonize across various collaborations. It's seeking blood trait data, which is something that has not been collected on many Africa CRFs. So this is a new challenge for how to discover other phenotype data that's not coming in on case report forms, but is needed for harmonization. So their intention is to pull their phenotype data together and harmonize it in a way similar to the CVD working group. So in our most recent meeting, the working group discussed how we could leave Africa in the best position to harmonize and share phenotype data. We had a really good discussion, and given the time frame that I have, I won't be able to go into all the details, but I'm just going to pick out a few. One of the key things that we identified was the need to make sure that when we publish the phenotype data and our recommendations for data harmonization, that we make sure that we publish and release them on all the right platforms so that they're clearly communicated. The other core thing that we identified was we still need to find a way to lower barriers to identifying commonly collected data across Africa studies. A number of members expressed difficulty in identifying other projects that have collected phenotype data that they could make use of, and they could approach those studies to make some collaborative research question going forward. It's still difficult to identify which projects have collected what data. And so that is a real challenge that we still need to work on addressing. Something that many members felt very strongly about is that it would be a real pity to lose the wealth of combined collected phenotype data simply because we don't clearly recognize and are able to identify what was collected by which project. So we really feel strongly about finding a way to repurpose that collected phenotype data. And so a lot of what our working group is looking at is how we can make sure that as much metadata about the H-Africa projects is collated and housed in a way that makes it accessible for sister organizations or other H-Africa projects to identify those projects they can work alongside. So there's still a number of months of work that's going into that. We did discuss how possible it would be to achieve a phenotype catalog similar to the H-Africa catalog and the IHCC atlas, but that could be an extensive amount of work given the time frame that we have. So our deliverables are mainly to focus on completing the current harmonization activities and outputs, communicate those clearly, and publish our recommendations for harmonizations and lessons learned. But we just wanted to raise that core kind of problem that we identify, which is that it's still a struggle to get H-Africa studies to share what data they have collected and to make that accessible in some way. So I just want to briefly acknowledge and thank all the active members. There are many people involved in phenotype harmonization activities and efforts, and I just want to give a big thank you to those who are participating and involved with us.
Thank you very much, Catherine. That was most interesting. We'll now go straight on to the next one, which is the microbiome working group, and the presenter is Voke Oduaran. Thank you.
Hello, everyone. I'll be presenting this report back on behalf of the Microbiome Task Force. Just to give a bit of an overview of the Microbiome Task Force, the idea of a dedicated Microbiome Task Force was conceived at the 14th H3 Africa meeting in Ghana. This was because of the great number of projects across the consortium with an interest in extending their current projects to include microbiome components. So we set out some goals for the task force, and we are a continuous work in progress. Generally, we aim to move quality microbiome research in Africa forward. To this end, we hope to apply lessons learned from global microbiome research, share knowledge and experiences as we learn across our various projects, and in so doing, build capacity to perform impactful research, as well as facilitate collaborative research and data sharing. This will obviously contribute towards the sustainability of the task force. So one way to work collaboratively is to have a project with consortium-wide interests. We thus set out to spend some time during the working group session last week to brainstorm on actionable projects of consortium-wide interest. And we agreed on a timely project that broadly investigates the role of the microbiome in COVID-19 immune responses. We also plan on asking several other questions, including the influence of vaccines. But we realized that the kinds of scientific questions that can be asked is dependent on the availability and accessibility of samples for research purposes, as well as the associated collected phenotypic data. We therefore decided to focus this report back presentation on our proposed study to get a sense of sample availability, both currently and potentially available samples within the next few months across the consortium. To accomplish this, we have, well, at least we hope to accomplish this by the poll we've created. It is intended to be a straightforward question with 5 responses to gauge the different levels of interest across, or rather amongst consortium members. So the question pretty much is, do you have samples that are available for COVID-19 research purposes? Choice A, yes, I do. Choice B, yes, I do, and I am interested. Choice C, no, I do not, but I'm aware of samples in my locality that are potentially available. D, I will be collecting some samples within the next few months, and I am interested. E, no, I do not, and I am not interested. Please kindly indicate your responses in the pop-up poll that are on your screens. It is appreciated. Also, just to point out that the intention is not to, by any means, infringe on independent project research questions. And that is why we'll be sending out a follow-up questionnaire based on the responses to this poll. I'll just give us a few more seconds to make our choices. Okay, thank you. I'll be happy to entertain any questions in this regard as well during the Q&A session afterwards. So finally, as a task force, we try to organize microbiome-focused webinars twice a year. We've had one in June this year, and it was a presentation by Professor Ami Bhatt from Stanford University. It was very well attended, thanks to all those that attended. The second one is scheduled for October 18th, and it will be a presentation by Professor Clement Adebamowo. We'll be circulating more information about it towards the, well, closer to the time. So please be on the lookout for our flyer towards the end of September. At this time, I would just like to acknowledge and thank all the members of the Microbiome Task Force for their resilience. I'd also like to say a big thank you to Francis and Ali for the excellent support they provide. The task force meets on the 3rd Monday of every month at 4 p.m. Central African time. You're welcome to join. Thank you as well to all those that participated during the working group session we had last week. Thank you.
Thank you. Thank you for doing the live poll. I hope as many people as possible responded. It'll be interesting to see the results of that. So now we're going on to the Education and Coordinated Training Working Group, and Pabalo Chawuke is going to present on this live. Pabalo.
That introduction. Good afternoon, good morning, good evening, everyone, depending on where you are in the world. Thank you for joining today's report back sessions. As Ruth said, my name is Pablo Chawuke, and I'm the current chair of the ECTWG, and I am co-leading with Professor Fozia Rudani as well, who might not be joining me today, but is somewhere in the room virtually as well. So I'm going to be reporting back on the items that we discussed as a group in the previous week, but also the things that we sort of achieved and plan to do in the next year of the grant that is left. So we spoke about mentorship program, which I'll explain now. Rolanda reported back on standard item metrics that we sort of used on HT Africa's, particularly fellows, but also all our training and education activities. And if anyone is interested in those slides, I can send them at a later point as well. We also are planning on sort of creating a questionnaire for evaluating training grants, and we discussed that as a group, coming up with sort of ideas on how to deal with that. One of the things that we've sort of realized is that mostly we focus on fellows training activities. So we're sort of like coming up with ideas on how to improve staff training, particularly for PIs and senior staff. We also brainstormed ideas around cohort exchange, depending on whether COVID allows or not. How are we going to have an internship / mobility program that we can sort of implement in the last year of a program, obviously pending COVID, as well as funding restrictions as well. We spoke about, so that was linked to the COVID exchange program. We also spoke about a prize for best paper or thesis, where students could sort of submit to universities their projects at the end of their degrees. But I mean, there was obviously a lot of feedback around this idea on how there would be a lot of challenges in achieving it. A few achievements that I'll mention is that we obviously, as I said, focus on fellows training activities. And in this current consortium meeting, we're going to be sort of coming together with the African Society of Human Genetics, HJ Barnett and other sort of institutions like Ensemble, etc., where we're going to be sort of training our fellows online for the next month of September. So please be on the lookout for those announcements as well. We had our members of a group review abstract and follow applications for this current iteration of the consortium. And we also have monthly meetings as well, and we have some manuscripts in the pipelines as well in the background. So just quickly to explain on the mentorship program, which is what we discussed last week, sort of Thursday, is that we need to create a program or a workshop where we can train some PIs and some senior staff and even young and up and coming early career researchers who want to be mentors on how to be mentors, because a lot of people sort of go to varsity but do not learn mentorship skills because it's not offered in degrees as well. So we should sort of, as a group, come up with ideas and ways of mitigating the shortfall as a group, because we're thinking of a more regional mentorship program where there's not going to be like a lot of money that needs to be spent, and this can be done online where a PI from one node can mentor one student from another node as well. So it doesn't have to be in the same sort of institution or node as well. So it has to be a sort of skill exchange as well as regional exchange in that as well. Rolanda, as I said, kindly and excellently presented on this, and there are slides on this where she sort of like had nice sort of graphs and stats on what we achieved as a group over the past sort of 10 years or so of the grant. And I think it would be a good, sort of even a paper written on this would be good, because it sort of traced the number of fellows, what they do, where they're from, what's the diversity amongst gender, country-wise, who are judges for presentations and stuff like that, and who contributes to our newsletters, etc. So that was like nice item metrics that she followed and nicely presented to our group as well. We were thinking of creating a questionnaire, and the group sort of came up with ideas on how do we sort of evaluate student progress, where we look at things like, when did they publish their first paper? Are they attending and presenting work at symposiums which are of a high caliber as well? We're looking at, for the whole project, what are the graduation rates of students? What are the outreach activities that we have? And looking obviously at sort of like trying to find grants as well as other financial attraction and collaboration as well. I'm looking at the time, so I'm going to speed up a bit here as well, because I have about like 3 minutes left. So for staff training, we come up with ideas on mentorship role. As I mentioned earlier, mentorship could be part of what PIs do and learn on how to do as well. Data visualization, critical thinking, problem solving. And again, we've offered this before, NIH grants administration, because not everybody sort of knows how to do this. So, I mean, it's quite important, and evaluation and feedback from before has suggested that this is a very important training activity for PIs and senior staff as well. With the cohort exchange, we're thinking about creating a similar program that programs like HJ Barnett have, where students can, for 3 months, or even, for us, we're not even going to just limit it to students, obviously pending on budget and on COVID as well, where students can move from Cape Town and Ghana for 3 months, obviously following resources and a particular supervisor and skill sets that they need as well. So it's something that we'll discuss, and obviously depending on whether we get funding and we have time, we will try and achieve this as a group as well. For best price paper, the feedback from the group was that this will be a bit challenging. So we are putting this or packing this in the background a bit and focusing on the other goals that I mentioned as well. And yeah, so I think I'm within time and I'm happy about that. And just to acknowledge that I don't work alone. I work with a team of people. I think Albert Einstein said, I stand tall because I stand on the shoulders of giants. There's a team of people behind me. There's a lot of us, about 50 of us in the education and training coordinated working group. And I just want to say thank you to every one of them, particularly Fozia Rudani, who's my co-chair. Rolanda, Julius, and Michelle Skelton, who are my anchors from coordinating center, as well as the whole team. Nikki Mulder, who's my manager at H3 Bionet, and the H3 Bionet team, who without them I couldn't do the many things that I do, as well as the funders. I will obviously as well be able to take questions and answer those at the discussion time as well. Thank you so much for the opportunity.
Thank you very much, Fabalo. Yes, excellent timing and very important point you made there about mentorship. Before we go on to the final three presentations, could I put out another word of encouragement to the attendees to post your questions in the Q&A box? We do have a Q&A session at the end, and I think all the presenters have mentioned key challenges that need to be addressed, and I'm sure you must have questions about those challenges as well as other things. Right, so without further ado, we'll go on to the Study Coordinators Working Group, and Sophia Osawe is going to present on this.
Good morning and good afternoon, wherever you are. My name is Sophia Osawe. I am the current chair for the Study Coordinators Working Group. And in the next few slides, I will be presenting our updates, especially since the last consortium meeting. So with me, I have David Nana Ajay, who is based in Ghana and part of the sickle cell group. And I'm based in Nigeria. I'm the study coordinator for the beaming study. With me, we have a strong support team at the H3 Africa Coordinating Center, Shumi, Rolanda, and Tanian, and we have support from Karen at the NIH. As part of our activities, we have monthly meetings, which happen on Zoom. This happened first week of every month, and we've been very consistent with those meetings. In terms of attendance, we have averagely about 10 to 15 study coordinators. This fluctuates. We also got new members since the last consortium meeting. So averagely we get about two to three study coordinators, new members of our team after every coordinating, after every consortium meeting. During our calls, we discussed on previous action items that we gave ourselves. We learned lessons from interacting with any project challenges faced, and we learn from each other. A key part of what we do is for capacity building. So we do ask study coordinators of any trainings or any training needs they may have, so that the coordinating center and Rolanda just look at that and see how we can get someone to come over to join us during our calls and train us. So that's what we've been doing in the past. So we've had a number of trainings in different areas like budgeting, but recently we had a very interesting training given by Rolanda on elevator pitch, and that was quite interactive during our working group meeting last week. And we will continue practicing that, those elevators pitches. Also, part of what we do, we started something last year to have like a journal club where we go over published papers that are more related to what we do. So the strategy that we used to make sure that study coordinators understand the importance of having journal club and to motivate us by showing videos on that. So we've done that during the working group meeting. One of our main activities and goal is to publish our manuscript. We've gone through a long journey towards it, and we're almost towards the end. What I'm showing here are just key areas of what we're developing right now. We are currently designing the questionnaire on REDCap, and Catherine is leading us through that. We're also putting together the consent form. We've worked on the literature review, and one of the areas we wanted to expand our manuscript is to touch the perspectives of all of us in terms of patient retention over the years, and what strategies we have implemented, and what are the key areas that we should focus on when talking or dealing with participant retention in our studies. As a way forward, we have a standing manuscript writing meeting, which started about 3 weeks ago, where we dedicate 1 hour as a team to expand the manuscript so we can write different sections. But in the last 2 meetings, we have finalized the questionnaire. So these are the key activities, and a lot of focus has gone into that over the months. I mentioned the journal club, which we have monthly during our calls. And one new part of what we want to start doing is the project highlights. This is being developed by the H3 Africa Coordinating Center, where we can have a study coordinator put in a write-up that highlights projects, achievements, and updates. Well, we all have challenges, and a few of our challenges are internet connectivity, because we're in different locations in Africa, where some may have poor connectivity than others. This is one of the reasons we moved from Uber conferencing to Zoom, and that is going much better. In terms of attendance to meetings, we have been working on improving that since last year. We put in place some strategies. One was the WhatsApp group that has been very active over the last few years, where we ask study coordinators to join, and we share quick updates on upcoming meetings or any activities that we have. We share papers, we share trainings. So that's really a quick information hub for us. The other thing is that we got some feedback from study coordinators that they were not receiving the group emails that we sent. So the coordinating center, few months back, I believe it's like 2 months back, sent a group email to the study coordinators, and we asked each of the study coordinators to let us know if they received it. So that was a great activity, and that has been rectified. On COVID-19 related challenges, these challenges are faced, I'm sure, by a lot of working groups and a lot of studies. We are learning from each other on that, as I mentioned earlier. But COVID-19 has affected us personally and in our careers. So we keep learning and moving forward. Thank you so much for listening to our updates. It has been an honor representing the study coordinating group and giving us the opportunity to share some of our activities. I want to thank our sponsors, NIH, also the great support team that we have at the H3 Africa Coordinating Center, and Karen at the NIH. And it's a pleasure working with a great team like the study coordinators. Thank you, and I'll welcome any questions you may have.
Thank you, Sophia. So we now have 2 presentations remaining, and the first of these is from the HIV/AIDS Working Group, and Nikki Tiffin is presenting this one. Thank you.
Hi everyone, I'm Nikki Tiffin from the University of Cape Town, and I'm reporting back on behalf of the HIV/AIDS Working Group. I will start with just a brief overview of some of the activities of the working group. Our first activity was to propose an HIV/AIDS meta-analysis with data from the different projects ongoing in H3 Africa. So we wrote a manuscript outline and we circulated that. But despite repeated attempts, we didn't get any buy-in from the H3 Africa projects themselves. So this project was eventually put on ice. The second activity that we undertook was to develop a framework for research, genomics research for HIV/AIDS, and we used the theme of HIV/AIDS through the life course. You may recognize the schema that we used, that we developed, and we used to guide our idea of what kind of genomics research we felt would be appropriate at the different life stages of our African populations. We then wanted to go forward and develop a written framework for research around this life course theme. So we used this as a basis for applying for supplementary funding to run a workshop where we could get together to come to a consensus on the research based on what we'd put together already, and then to write a white paper or research outline for HIV/AIDS genomics research in Africa and the way forward, a kind of consensus way forward. We were granted the funding, but unfortunately, due to COVID-19, we've had to put the workshop on hold. And then also at the beginning of the pandemic, particularly, a lot of our infectious disease experts in the working group were really caught up and remain caught up in the COVID-19 response. So because we had very poor attendance and people were so caught up elsewhere, we actually put our meetings on hold for quite some time. When we restarted our meetings, it was apparent that we weren't going to be able to meet in person for quite a while yet. So we decided to start to write the white paper prior to the face-to-face workshop and do this as a kind of online collaborative effort. Again, addressing HIV/AIDS genomics research through the life course. So our meetings resumed. We prepared an outline of this white paper, which was circulated. And then we also engaged potential collaborators in this writing exercise from outside H3 Africa too, and identified section leads for each of the sections in the white paper. Unfortunately, however, we're still encountering problems with poor meeting attendance. We've really made no progress on the white paper, and we also have failed to get any volunteers or candidates to co-chair alongside the chair position. And we think this is probably because people are very, again, still very involved in the COVID-19 response, and maybe infectious disease experts don't have the bandwidth currently to engage with this fully. So we've decided to put the working group back on hold until we can actually run the face-to-face workshop, which will be a more immediate way to engage together to take this forward, and also will take place once the worst of the pandemic is behind us. Thank you.
Thank you for that presentation. It sounds disappointing that you've had all these challenges because of COVID, etc. But let's hope that when you can run the workshop that things will take off again. Okay, so now we will have the presentation that was originally scheduled to be near the beginning of this session, and it's the Rare Disease Working Group, and the presentation will be by Aimé Lumaka.
Hello, everyone. I'm happy to present the contents and the discussions of the H3 Africa Rare Disease Working Group session on behalf of the members of our working group. So last Tuesday, we all held our working group session for this 18th H3 Africa Consortium meeting. Our meeting had 4 main agenda items. The first one was an update on what is going on within our working group. The first one is the engagement with the CleanGen. We have 1 monthly call, and during those calls, we discussed around rare diseases, and we are now exploring the way to extend this collaboration to the NIH Mendelian centers. The second update was given about the manuscript we have written a manuscript on the usefulness of increasing diversity in data repositories and the impact of this on the diagnosis and the care for rare disease patients. And this manuscript was submitted for opinion prior submission to the Orphanet Journal of Rare Diseases. The group decided that a small group of people will meet to review the edits and ensure that the manuscript remains within the scope for which it was primarily written. The third update was about capacity building. We are organizing a hackathon on data interpretation for rare diseases, and we are also planning to hold a workshop on an application that allows users to capture the pedigree. During the discussions in the working group session, we realized the overlap or the similarities with the GA4GH effort to devise guidelines for pedigree. And two members of our working group, Mamana and Judith, will design a concept document that will frame the collaboration that we would have or might have with the GA4GH for this activity. Rolanda from the H3 Africa Coordinating Centers will help to create a link with the fellows so that those activities, training activities we are developing within the H3 Africa Rare Disease Working Group would be beneficial to the trainees, offer an additional training opportunity to them. The fourth one was about the survey of existing capacity for rare and Mendelian diseases in Africa. The idea is to build a catalog for those who are looking information to be able to retrieve them much more easily. Jerry, he's preparing a survey design, and other members have offered to join and help her. The last one was a spontaneous expression from members who attended our meeting but are not already part of any H3 Africa project. And Rukaya Ndiaye from Senegal expressed her enthusiasm and positive feeling about being part of this working group. The second agenda item was a testimonial of people from the H3 Africa that are attending the ClinGen activities. So within the H3 Africa, the DDD Africa especially is strongly involved as they see this as an opportunity to make variants public and therefore increase the diversity in existing databases. And for the DDD, this is also a way to explore barriers to submission, why people are not submitting that much information to the public repositories. Amanda and Emil have joined the bio-curator for the Gene and Curation Expert Panel on Intellectual Disability and Autism. Overall, the feedback is very positive, and those representatives are viewing this as an opportunity to develop critical approach for the evaluation of functional studies and assessment of gene and disease association. For the third agenda item, we all visited the results of the follow-up poll that was designed to capture the involvement and the implementation of skills and knowledges that people acquired during the joint workshop that H3 Africa Rare Disease Working Group co-organized with the ClinGen in February 2021. At the time of the session last week, 16 people responded to this poll, and they were from 9 groups, mainly from the leaders group from Mali. And among them, 8 of them are involved in a way or another with the ClinGen. The reason is because they are willing to receive training to enhance their knowledge, gain experience with ClinGen resources, and also learn how to share resources and ideas. But for those who are not yet engaging with the CleanGen. The main reason is that they don't have enough information to make an informed decision. Interestingly, for those who attended the workshop and are not practicing the knowledge they gain on a daily basis, the main reason was the lack of data. People reported that they are not yet in possession of data that will allow them to use the CleanGen tools and practice what they learned during the workshop. We heard from representatives from Rwanda, Senegal, Nigeria, and Mali that the lack of data and the limited bioinformatics skills are the two major barriers. So the group concluded that there is a need to create artificial data simulated from real cases, and the need to regularly invite experts on our calls so that they can bring additional information that would help people to make informed decisions. And the group encouraged members to attend practical training from the H3 Africa Bionet. Last point was about the planning for the hackathon. This is intended to be a hands-on session for NGS data mining for rare diseases. And volunteers are invited to collaborate and express the area of interest where they would like to bring contribution. The idea now is to couple the hackathon with a training course by the H3 Africa. So the group observed that the best way to allow people to practice what they are learning and to capture the most of this hackathon is by providing them with the theoretical knowledge that will prepare them to the hackathon. A brainstorming session is going to be organized in the upcoming days that will put together members from the H3 Africa Working Group, the H3 Africa Bionet, the CleanGen, and the NIH Mendelian Centers. Going to the hackathon, a survey will be made available to capture the needs for skills and training. PIs are warmly invited to inform the working group about what they are missing in the teams and what they would like to add to the team. Thank you very much.
Thank you very much. So that completes the presentations from the working groups, and we're now going to move to the Q&A session. Now, we have some questions that are directed at specific presenters and some questions that are addressed to the whole group. So I'm going to start with the ones that are addressed to specific people, and then when we've done those, we'll go to the ones that are addressed to everybody, and we can go around the panel and see what people would like to say in response to those. So take the first question. This is for Catherine Johnston, and it's from Gunther Jensen. And the question is, data harmonization is perhaps one of the most daunting challenges. I was very enthusiastic to learn how much progress you have made. Does phenotypic harmonization for data from African participants come with additional specific challenges that go beyond those already known from previous biobanking and sequencing efforts elsewhere? For example, a broader spectrum of diseases, more detailed environmental data models, etc. Catherine.
Thanks. Thanks, Ruth, and thanks, Gunther. I'm not sure if I fully... Hi, can everyone hear me? There was a bit of a blip there. Hopefully I can be heard.
Yeah, you are audible, yes. Yes.
Sorry, just the screen went blank. Yeah, thanks for the question. So I'm not sure if we've clarified exactly what those specific challenges already known are, but I can just speak to the challenges that we have discovered along the way. Obviously, the phenotype data sets involve a lot of heterogeneity. They're very different, and people ask sometimes the same question in many different ways, and sometimes they ask different questions in the same way. So it does come with many challenges to find a harmonized common data model. So if you're wanting to join the data together, that does present the immediate challenge. And obviously we have tried to work to resolve that in the H-Africa Consortium by firstly identifying some common phenotypes that everybody agreed to collect, and then continuing that work into a more refined harmonized data model. But it does come with additional challenges, specifically in our continent. I think many prior phenotype harmonization activities often occur within the same country. And so in our context, we are often dealing with multiple countries with different government policies and ethics groups governing the data that has been collected. So it does come with careful reviewing of what participants have consented to in the first place to make sure that what we're doing is first and foremost ethical. And so that does start the process of trying to harmonize the data. And then there does, in the context of which we've been working, we have found that there is hesitancy to share the phenotype data that has been collected. I'm not sure if we have understood fully the barriers to sharing what data has been collected, but that has been a major struggle for us to assist the projects to feel comfortable to share their metadata about the phenotype data they collect. And I think part of that comes with perhaps most of the harmonization efforts that take place start off with an agreed upon collaborative research question that they're trying to address. Whereas this consortium has formed to address genotype data collected collaboratively and submitted to the EGA. I think perhaps the phenotype data did not come with a clearly defined set of rules for collection and collaboration to start with. So I think that has kind of set the scene for difficulty in projects to know what they should share and how they should share it, and why they are collaborating on this work. So I don't know if that's answering the questions. If there are further questions, I'm happy to address them.
You've raised a lot of issues there, Catherine. It was most interesting, and I think we could go on discussing that issue, but I think we need to turn to some of the other questions. So thank you for that. The other question to an individual I have is for Pabalo. It's from Zane Lombard, and says it would be really interesting to know how many junior consortium members and fellows have gone on to successfully obtain funding for their own research. And is this something that your working group is looking at? But before you answer that, Pabalo, I just want to say to all the attendees, it's been brought to my attention that not everyone may be able to find the live Q&A chat, and you need to press the green button on the bottom right of your screen, because the live Q&A chat is in the AirTouch device, and there's a button that you can press. So I hope that's helpful, and sorry to delay you in your answer, Pabalo. Would you like to give it now?
Thank you so much, Ruth. Thank you to Zen for your question. And I think I'm going to note that down, that I don't think we've actually looked at that yet, but I think it's something that we should definitely. What we do as the working group is that we offer training on grant application. So we then need to sort of now find out or evaluate if sort of our training is having any impact. So I think it is a very great idea, and I'll jot it down. Thank you so much for your question, Zane.
Thank you very much. So now I'd like to turn to a question from Jennifer Troyer, and it's to all members of the panel, because she's noticed that buy-in is a recurring theme. I mean, we've heard a lot about not being able to get data from different projects and so on. And so she says, realizing there are many challenges, what is needed to gain enthusiasm and buy-in for meta-analyses? Is there just no interest, or are there barriers we can address to move some of these things forward? So who would like to go first? If no one volunteers, I'm going to pick somebody. Okay, right, let's see. What about Sophia?
Okay, thank you. I was thinking probably the question will go to a group that is really having a hard time with that. But in terms of my opinion, I think this is something we should continue discussing about. I'm not sure it's a challenge that we have as story coordinators, but definitely this is something that we should put as part of our agenda in our working groups to discuss among ourselves what are the barriers to having groups buying into meta-analysis. And definitely we can find some key points that will come out, or some key ideas and strategies that we can implement as groups to make sure that, you know, we overcome this barrier. So personally, I think we can.
Okay. Thank you very much. What about Bamidele?
Yes, I think first of all, the issue can partly be addressed from the research group level, because sometimes the question is, how does this actually advance the sustainability of the research group? Because each research group will want to advance their study objectives and then be able to sustain themselves. Because sometimes, okay, we talk of meta-analysis or a project, you know, manuscript and all the rest. Then if a research group does not see an advantage or an option for leveraging, then it becomes difficult for them to really, you know, jump on it and participate. So I see it from, first of all, being able to convince the research group that there's a lot that can be gained in participating.
Thank you very much. I have a message from MA that MA would like to contribute on this topic.
Yeah, thank you, Ruth. My working group, the rare disease working group, is one of the group that has struggled with these since months now. And some of the ideas we had couldn't go through because of some issues in data sharing. It could be very easy to ascribe that to a type of culture, say that people don't have the culture of sharing, but a deep analysis may show that it's just people didn't anticipate that this would come one day when they were designing their own project. That's why the design of the project, the way data were collected, this was not done with the idea of multi-purposing collection, that you may collect the data and the de-identified data from your project may serve in another project. You may collaborate this data with others. And in another illustration, this was even not considered in the consent in many projects. So now when we are knocking on the doors asking for people to collaborate with the data, we are stuck with this issue. They didn't obtain the proper consent. I think a way to do this is to consider a second level consent where the IRB would provide consent for using the identify and summary statistic data to advance research. Otherwise, those data would not be useful beyond the original project. I think it was a problem with the design, and that we can resolve that if we consider the opportunity to use the IRB as the second consenting for de-identified and summary data.
Thank you very much. I've also had a message from Michelle Skelton that the H3 Africa Coordinating Centre is trying to address this issue with surveys and interviews in the coming months. Other people have indicated willingness to contribute on this topic, but as we've only got 4 minutes left, I think we probably need to go on to another question. And this is a very general question, and it's tied in with the last one. And it's from Ali Osgood. In what ways can consortium members help advance the goals of your group? In what ways can people help if they're unable to join the regular working group meetings? And I think I'm going to combine this with another question, which is anonymous, which is asking in what ways younger investigators are encouraged to participate and have opportunities for learning and leadership in your working group or task force. So those are two general questions, one about the ways in which younger investigators can contribute, and others, what help you might think could be useful to you from members of the consortium in achieving the goals of your group. So I'm going to ask people who haven't yet contributed to the previous question if they would start on this. Nikki.
Thanks, Ruth. Yeah, I'm happy to comment. I think they are all quite tied together. As I outlined, the HIV/AIDS working group has really struggled to engage. And I don't really have an answer specifically to the questions. But what I can say is that one of the approaches we tried to use for the HIV/AIDS working group was to ask PIs from the different studies to identify maybe a younger, early or mid-career scientist from their project who they thought might like to lead their participation in the working group. Because we thought that, we recognize PIs are often very busy and may not have time to engage directly. So even that wasn't successful. So to be honest, I'm not really, I don't know where the problem lies. If the problem lies that the message doesn't reach the younger and mid-career scientists, or whether they decide that they don't have the bandwidth perhaps to take things on. So I think I'm coming back more with a question than anything is, where does this, and I guess I can put the question to also our audience where there must be young and mid-career scientists. Is there an interest in engaging and representing projects in the working groups? I'm not sure where the block comes from, in fact, because we certainly used that as a strategy unsuccessfully to try and engage further. So also to answer the question of what can the rest of the consortium do, I think it's about making sure that when these invitations go out, the invitations to participate in the specific projects actually reach the people to whom the message should be going. And then, to encourage and support younger scientists to engage and maybe offer to mentor them if you don't have, the time yourself to join the working group, to be kind of a backstop for younger scientists getting involved. I think that could be very helpful. Thanks.
Thanks, and very useful comments there. Okay.
Hi, thanks, Ruth. I think I would just comment on Alice's question about how people can help if their consortium members can help if they are not really a part of or they don't really have the time to attend the working groups, the working group or the task force sessions regularly. I think one thing is that the messages are sent out, the announcements, the meeting notifications are usually sent out during to mailing lists. And the minutes of the meetings as well, in our case, in any case, is also usually sent out to the mailing list. I think just responding or indicating an interest, and if for some reason you can get in touch with one person in particular, perhaps the H3 admin is always, I think, very good with getting us in touch with people that are looking to connect. So I think that's one way, just interacting with the H3 admin, responding to topics of interest or points of interest, and just letting us know that, at least letting whatever working group know that you're interested in a certain part of their discussion or project, and how you propose to support, or just open the lines for further communication. I think that's a very good way of getting in touch with the mailing lists. Thank you.
Thank you very much. Well, we are over time now. I'm very sorry if someone feels they have a comment they want to make, which they haven't been able to make, but it's still possible to answer questions after the session, of course, by virtual means. So I want to thank all the presenters and all the attendees at the session and the backup support for the technology. It's been a most interesting session, and thank you once again, and goodbye for now.